Greetings to the viewers. In this video, we are going to see about the FDA-approved gene therapy for children with metachromatic leukodystrophy. Lenmeldy, atidasogene autotemcel, is the first gene therapy approved by the FDA for the treatment of children with metachromatic leukodystrophy, MLD, that is presymptomatic late infantile, presymptomatic early juvenile, or early symptomatic early juvenile. First, let us see about the disease. Let's break down the term metachromatic leukodystrophy into its components. Meta, this prefix comes from Greek and can mean beyond or change. In medical terminology, it often indicates a change in condition or state. Chromatic, this term relates to color. In the context of biology or medicine, it often refers to staining characteristics, particularly in cells or tissues when viewed under a microscope. Leuko, another prefix from Greek, it pertains to white or white matter. In medicine, it's commonly used in terms related to the white matter of the brain and spinal cord. Dystrophy, this word comes from Greek roots meaning faulty or abnormal nutrition. In medical terminology, it typically refers to a disorder characterized by inadequate or defective nutrition or development of cells or tissues. Putting it all together, metachromatic leukodystrophy refers to a genetic disorder characterized by a change in the staining characteristics of certain tissues, metachromasia, and abnormal development of white matter in the nervous system, leukodystrophy. This condition leads to progressive deterioration of neurological function. There are three forms of metachromatic leukodystrophy, which involve different age ranges: late infantile form, juvenile form, and adult form. Signs and symptoms can vary. The infantile form is the most common and progresses more rapidly than the other forms. Metachromatic leukodystrophy is an inherited disorder caused by an abnormal, mutated, gene. The condition is inherited in an autosomal recessive pattern. The abnormal recessive gene is located on one of the non-sex chromosomes, autosomes. To inherit an autosomal recessive disorder, both parents must be carriers, but they do not typically show signs of the condition. The affected child inherits 2 copies of the abnormal gene, one from each parent. The most common cause of metachromatic leukodystrophy is a mutation in the ARSA gene. This mutation results in a lack of the enzyme that breaks down lipids called sulfatides that build up in the myelin. Rarely, metachromatic leukodystrophy is caused by a deficiency in another kind of protein, activator protein, that breaks down sulfatides. This is caused by a mutation in the PSAP gene. The buildup of sulfatides is toxic, destroying the myelin-producing cells, also called white matter, that protect the nerves. This results in damage to the function of nerve cells in the brain, spinal cord, and peripheral nerves. On March 18, 2024, FDA approved first gene therapy for children with metachromatic leukodystrophy. Lenmeldy is a one-time, individualized single-dose infusion made from the patient's own hematopoietic, blood, stem cells, HSCs, which have been genetically modified to include functional copies of the ARSA gene. The stem cells are collected from the patient and modified by adding a functional copy of the ARSA gene. The modified stem cells are transplanted back into the patient where they engraft, attach and multiply within the bone marrow. The modified stem cells supply the body with myeloid, immune, cells that produce the ARSA enzyme, which helps break down the harmful buildup of sulfatides and may stop the progression of MLD. Prior to treatment, patients must undergo high-dose chemotherapy, a process that removes cells from the bone marrow so they can be replaced with the modified cells in Lenmeldy. The safety and effectiveness of Lenmeldy was assessed based on data from 37 children who received Lenmeldy in 2 single-arm, open-label clinical trials and in an expanded access program. Children who received treatment with Lenmeldy were compared to untreated children, natural history. The primary efficacy endpoint was severe motor impairment-free survival, defined as the interval from birth to the first occurrence of loss of locomotion and loss of sitting without support or death. In children with MLD, treatment with Lenmeldy significantly reduced the risk of severe motor impairment or death compared with untreated children. All children with presymptomatic late infantile MLD who were treated with Lenmeldy were alive at 6 years of age, compared to only 58% of children in the natural history group. At 5 years of age, 71% of treated children were able to walk without assistance. 85% of the children treated had normal language and performance IQ scores, which has not been reported in untreated children. In addition, children with presymptomatic early juvenile and early symptomatic early juvenile MLD showed slowing of motor and/or cognitive disease. The most common side effects of Lenmeldy are fever and low white blood cell count, mouth sores, respiratory infections, rash, medical line infections, viral infections, fever, gastrointestinal infections, and enlarged liver. After infusion with Lenmeldy, patients should be monitored for neutrophil counts and risk of delayed platelet engraftment until engraftment has been achieved. Treatment with Lenmeldy may be associated with formation of blood clots or a type of swelling of brain tissues known as encephalitis. There is a potential risk of blood cancer associated with this treatment. However, no cases have been seen in patients treated with Lenmeldy. Patients receiving this product should have lifelong monitoring for hematologic malignancies, including a complete blood count, with differential, annually and integration site analysis, as warranted, for at least 15 years after treatment. The application received priority review, orphan drug, rare pediatric disease, and regenerative medicine advanced therapy, RMAT, designations. The FDA granted approval of Lenmeldy to Orchard Therapeutics.